Good afternoon. It's our pleasure receiving you all today for our second day of our webinar of COVID-19 in immunocompromised group. The subject today is treatment, and I have the pleasure to introduce our two lecturers. The first one is Professor David Van Duyn. Professor David is an assistant professor at the University of North Carolina, and David will talk about convalescent plasma and IL-6 and antibodies. And after that, we will receive Professor Esper Callas. Professor Esper is a full professor here in São Paulo at the University of São Paulo, and Esper will talk about Remdesivir and others antivirals. David, thank you very much for your participation, and you can share your presentation, please.
All right. So I just want to thank the organizers for giving me the opportunity to present here today. It really is my great honor and my great pleasure to do so. So I'm going to talk about the use of convalescent plasma, IL-6 blockers, and antibodies in the treatment of COVID-19. Here are my disclosures. I'll leave these up for a second. So I will just start with a thank you slide. There are lots of people to thank at UNC. These are some people that have contributed to the work that you're going to see on some of these slides, and also on various studies that we've been doing at UNC, as well as in the clinical care of patients with COVID. So we'll start by talking about convalescent plasma, and I think for a lot of infectious disease doctors, the COVID pandemic was sort of a reminder of what plasma is again exactly. So I just wanted to put up this slide to remind everybody of what is in there. Obviously, we're mostly focused on the antibodies that are in there, but there's many other components, as you can see here. And then that antibody mixture is also a heterogeneous mixture of different antibodies, and those antibodies also vary from donor to donor. So in the top figure here, you can see 3 donors just shown as an example, and then what they what percentage of the antibodies are either binding to the spike protein and or the specifically the receptor binding domain of that spike protein. And you can see that much smaller percentages of those antibodies are actually neutralizing. So some early data came out, and this is one of the first randomized controlled trials that looked relatively promising from China to show that there would be a more rapid clearance of viremia with the use of convalescent plasma. And with that data, and also, of course, with a lack of available treatment options, the use of convalescent plasma really skyrocketed in the US. And in a lot of places, including at the University of North Carolina, the use of plasma really took off, and a lot of people received this therapy. Now, there's been a fair bit of criticism that while 80,000 patients received this therapy, much smaller numbers have been enrolled in trials in the US. And while I think, of course, that we do need trials and we should try to get as many people into trials as possible, I do think that we have to put this into context of having, again, very few options available, and most hospitals setting up a plasma program without external funding. So it's not quite as simple, I think, as sometimes it's made out to be. So there's a lot of observational data on the use of plasma, and I'm just showing you here one example that was relatively well done with a propensity score matched control cohort. And as you can see, there seemed to be a mortality benefit here on the red line on patients who received convalescent plasma as compared to matched controls. And when they looked sort of in a subgroup analysis, this benefit was primarily seen in those patients who were not intubated and who had shorter duration of symptoms. So the theme that was starting to emerge is that if plasma is going to do anything, it is probably going to be useful early on in the disease in patients in whom severe disease has not yet developed. And of course, that makes sense sort of from the pathophysiology in that if you are depending on blocking the virus, you want it to happen early in disease before the whole immune response is sort of unleashed. So here's an overview of what we've done at our institution, and these efforts have been led by Dr. Luther Bartelt, who has really been working tirelessly on this effort. So the first phase was just to get it started, to get the logistics, to make sure we were getting donors in who had COVID. And of course, all of this was no small feat, and the work from the blood bank was extremely important in that respect as well. Then the second phase we're currently in, and that is we've set up a small pilot trial, and I'll talk a little bit more about that, called the COVID trial, in which we're comparing high titer versus standard titer plasma for the treatment of COVID-19. And that's in hospitalized patients. And then as a third phase, we're really interested in diving into the mechanisms of action of plasma and how these things work, and trying to really dissect which antibodies in this heterogeneous mixture are helpful and how that might work. So talking about phase one, the implementation, we started our first donation in April, and from then until September, you can see the numbers here. So really lots of units collected in that time, and again, lots of credit to the team that has made all of this possible. And when we look here at those donors, we see that the distribution of blood types was very similar to what you would expect in the general population. So about 40% each of blood type O and A, 10% of B, and then only 6% AB. And I've just put in, again, as a reminder for most ID docs who may not have this sort of at their fingertips, of what kind of plasma you can give to what kind of patients. So patients with blood group A can receive A or AB plasma, etc. So looking at characteristics of the first 100 or so donors, again, this theme of heterogeneity is very clear. So we've done a lot of really in-depth characterization of the plasma that is donated and looking for various antibodies using various assays in different labs. And what you see is that some donors have very high titers on some assays. Some donors have very low assays, some are in between. So it's really all over the place. So to talk about the efficacy of plasma is sort of a misnomer. You really want to characterize as much as you can what you're actually giving. And I think those insights will also be helpful as we move sort of to the next phase of monoclonal antibodies. And in the pie graph here, you can see that in about 75%, these three assays were all positive as far as which antibodies were found. So here's then the overview of the so-called COVID trial. Again, it's a pilot trial with 56 patients. They'll be randomized 1-to-1 to either getting a standard titer, which we defined as 1-to-160 to 1-to-640, or a high titer of over 1-to-640. And the objectives are really to look at safety, the efficacy, and also virologic outcomes after giving plasma. And we're trying again to get people in as soon as possible. Now, of course, when you do this in a hospitalized population, as we are doing, you're always limited by the fact that it takes people time to come to the hospital. And also by that time, disease tends to be relatively severe already. So I just want to go over some trials that have been done with plasma. Now, the first one I'll mention is the plasma trial from Argentina. This included patients with severe COVID-19 who had a median of 8 days of symptoms. Severity was defined by an O2 sat of less than 93% on room air and or a SOFA score of at least 2. The majority of patients were on the ward, about a third were in the ICU, and only a few were in the emergency room at the time of randomization. They were randomized 2-to-1 to either receive convalescent plasma, and these were titered. So there was another trial that I won't go into from India, where they used untitered plasma, but given the heterogeneity that I've already told you about, I thought that would be less useful. So the total antibody titer here was greater than 1-to-800, and these were compared to placebo. The primary outcome that was evaluated was the clinical status at 30 days, and this was an ordinal outcome, with death being the worst outcome and discharge back to baseline being the best outcome. And then they had these levels in between that you can see here. They enrolled over 300 patients. About 200 of them were randomized to plasma, 100 were randomized to placebo. And additional anti-COVID therapy, which I put into quotation mark, and then the next speaker will, I'm sure, go into that, are listed here. So this was a negative trial. No effect of plasma was seen, and I'm showing here the distribution in these various groups. So in red, you can see the patients who died, so that was pretty much the same in both groups. And then you see the distribution of patients who remained on mechanical ventilation, et cetera, et cetera. And then the largest bar at the end in dark blue are those who made a full recovery by day 30. So the next trial I want to discuss is the trial that was also done in in Argentina. And this was trying to give the plasma earlier in disease. As I mentioned already, the biologic rationale is that the earlier you give it, the more likely it is that you'll see a benefit. This is only on preprint, so it's not undergone peer review yet. It was also a double-blind trial, and they enrolled elderly patients either over the age of 75 or those over the age of 65 with at least one comorbidity. And they had to have mild COVID-19 with less than 72 hours of symptoms. So they really got to these patients really quickly, and the author should be really commended on carrying out this trial. Again, randomization here was a one-to-one randomization to either plasma or placebo. And the primary outcome here was progression to severe COVID-19 before day 15. And severe COVID-19 was defined as having a high respiratory rate or a low O2 sat. They enrolled 160 patients distributed evenly between plasma and placebo, median age was 77, and they were pretty much equally divided between those two groups that they were enrolling in the age group. And most patients had at least one comorbidity. And here they did see a signal, and that plasma was associated with a reduced risk of progression. In the intent to treat analysis, the relative risk was 0.5, so pretty good effect size. And in this table 3 shown here below, you see that effect size was highest in those with higher antibody levels. So moving from plasma to monoclonal antibodies, where of course there's been a lot of activity, and just recently 2 of them have been approved under an emergency use authorization by the FDA here in the US. So bamlanivimab is a recombinant protein against the spike protein, and you can see that IC50 there. This was provided in the package insert, frustratingly, in microgram per ml. So I needed to get my daughter in with her up-to-date chemistry knowledge to convert it to picomolar. So if there's a mistake there, then let her chemistry teacher know. The other combination that is also under the same EUA is this casirivimab and imdivimab, which are given together. They're 2 different antibodies, both directed against the RBD of the spike protein, and you can see the IC50s listed here for in isolation or when given together. So these are the trials that led to these authorizations. So there's one study for each. This one was published in New England Journal, the BLAZE-1 study, which is really an interim analysis of a phase 2 study. So pretty, fairly preliminary data, I would say. This was done in the US, 41 centers in ambulatory patients who had mild to moderate COVID-19. Adults were included, and they had to be included within 72 hours of the positive test. So they could have symptoms for a longer time, but the test had to be within 72 hours. So it was a 4-way randomization to various doses of this antibody versus placebo. And the primary outcome was a virologic outcome, and that was based on NP swabs between baseline and day 11. About 460 patients were enrolled, and you can see the distribution here. So about 100 in each of the antibody groups and about 140 in the placebo group. So these were younger patients, about 45 was the median age, 4 days of symptoms, and with high viral load at baseline with cycle number of 24. So when we look at the virologic outcome, the primary outcome was really only met for the 2,800 milligram dose. But they describe in the FDA document that they figured that there was a flat dose response curve. So they went with the lower dose instead, with the 700 milligram dose. And of course, the point estimate is very similar. And there was also an effect seen on the hospitalizations. But these numbers, of course, were very small. Only 9 hospitalizations in the placebo arm, and 1 or 2 in each of the treatment arms. So the data on these other 2 antibodies comes from one phase 1/2 trial, and these are unpublished data. So all of this comes from the FDA fact sheet. This is a double-blind randomized controlled trial, also on ambulatory patients with mild to moderate COVID-19, again within 72 hours of a positive test. These patients were randomized 1:1:1 to 2 doses of combination antibody versus placebo. And again, a virologic endpoint was used. And here they used a time-weighted average change between day 1 and day 7. They had about 800 patients enrolled, and you can see here the distribution, pretty even between the antibody groups and the placebo group. Again, relatively young patients of around 40, 3 days of symptoms in this in this study, and the viral load was about 5.8 at baseline. So you could see a difference between placebo and all antibody groups, and no real difference between the 2 doses. And again, also hospitalization or emergency room visits were also lower in the treatment groups. Again, note the small number of events. So switching gears to the IL-6 blockade. So as I've taught you about the plasma and where the plasma makes sense to give it early on in disease, IL-6 blockade and other immunomodulators really make sense later on in disease, where the virus has sort of played out its role, and now you have an immune system that is out of control and is likely causing most of the damage. So very early on, this hyperinflammatory state was recognized, and people started comparing this to a cytokine storm similar to what you might see in CRS. And a variety of articles came out, and this idea that IL-6 played a central role in the cytokine storm took a foothold. So the idea that treating with IL-6 blockers that might have a positive effect made some sense. And looking here sort of at the biology of where that could have a good effect, you see here where these various blockers work. It's important to keep in mind that IL-6 leads directly to the release of CRP by the liver, and of course we often measure CRP clinically, and CRP is often elevated in these patients with severe COVID. So at UNC, we were very, I think, similar to other centers in that we started to give IL-6 blockers in the absence of good data in patients where we felt that our back was against the wall. And there was an in-depth look at this under the leadership of Dr. Parr, shown here at the top. And what you see is that the CRP, as expected, normalized quite rapidly. But then when you look at clinically more relevant issues, there was really not a consistent pattern of benefit. And then a lot of observational trials have been published, and a number of trials, and this is a very nice overview of that literature, also by Dr. Parr in JAMA Internal Medicine. And really, when you zoom in on these randomized controlled trial results, for the most part, they're all negative, and there's certainly no mortality benefit, leading to really the recommendation that this is not supported for routine use. So in conclusion, if you're going to use plasma or any other antibody-based therapy, you want to give it early on in disease when there is less severe disease and when the virus is playing the most important role. There's no definitive evidence yet for the efficacy of convalescent plasma. It is important to keep in mind that giving plasma and giving plasma might be two entirely different things, that it's a very heterogeneous product, that the concentrations of antibodies are going to be very different from one donor to the next. And even if you use the same donor on two different time points, you may get very different results. The knowledge that we gather from this experience with plasma, I think, is really exceedingly important in general for our understanding of the immune response to COVID-19, but also for more directed therapies such as monoclonal antibodies. And in contrast, if you're going to modify the immune response, then that is obviously indicated later in disease. And I think the best example of benefit is with dexamethasone. Unfortunately, IL-6 blockade has really not lived up to the promise so far, and is not indicated routinely. So I'll stop there, and I think we're doing questions at the end of this session.
Thank you very much, David, for this excellent and very clear presentation. We will have time at the end of the second lecture for questions and discussion. And now I have the pleasure to introduce to call for his lecture, Professor Esper, please.
So yeah, hi. It's a pleasure to me to be here tonight, this afternoon, to talk about more of our experience on monoclonal antibody development program in clinical research that we have here. But so unfortunately, I'll have a little bit of an overlap with the previous presentation. I apologize, but my idea here is to deep dive into monoclonal antibody usage for COVID-19. Obviously now, every time I present any lecture, I usually put the date, because probably by the end of this week, it's going to be outdated. So as far as monoclonal antibody goes, it's a long use in medicine. We've been using this kind of product since 1986, when Monab reached the market. Was licensed to be used to treat kidney graft rejection. The name is orthoklone, and those who are more old like me recall the problems using this medication, but it improved the treatment for rejection in these kind of grafts. A little bit of nomenclature on monoclonal antibodies, so people can understand when they look at their names. We have here a prefix that usually have in the middle of the word the target for the monoclonal antibodies for infectious diseases or immune-mediated diseases. In here is the virus. You're going to find the VI in the middle of the word. In this suffix will refer to the origin of this monoclonal antibody. So you have the first generations. We used to have most of them coming from other animals, and then they start being chimeras, and finally they start getting humanized. And the most recent generation of monoclonal antibody products are fully human, which increases its ability to be used in humans due to low toxicity, high tolerance, and better lifespan, half-life. I apologize. So let's go back to the coronavirus. The SARS-CoV-2 virus is here. Just remind you all that the spike protein buds out to bind to the ACE2 receptor on the surface of the cell. It's one of the four structural proteins for the virus, but I would call your attention that the RBD, which is located in the tip of the spike protein, is the most and the best target for antibodies to neutralize virus replication. In this case, if you look from upwards, you're going to see that in green here, the RBD, the receptor binding domain, where the virus, most of the monoclonal antibodies, they are most effective against SARS-CoV-2 bind, but also the S2 region of the protein is also a good target for the binding of these monoclonal antibodies. So the basis is it's a beautiful slide that Barney Graham sent to me a while ago. So showing the spike protein on the surface of the virus, the ACE2 receptor, and here's the binding for the infection to take place. And what comes along is when you use the monoclonal antibodies here, you can block this step of this infectious synapses, therefore neutralizing the virus. So our antibody is important for protection against SARS-CoV-2. There's a lot of debate about this, but the entire literature is converging to demonstrate that potentially humoral response is one of the best surrogate markers of protection, if not a true mechanism of protection against the infection and but mostly disease development. So here is just recall you all this New England Journal paper from the Icelanders who followed about 10% of the entire population in a cohort and found those who developed COVID-19. The vast majority had antibodies that neutralize the virus, but also binds to the RBD. And these antibodies were present throughout, down along 4 months after the infection took place. Another paper coming from the US now, from the New York region, followed almost 6,000 community members who had COVID-19 cases, and all those who seroconverted had confirmed by a positive PCR, had 2 weeks later antibodies against 3 regions of the virus, the RBD, the S2, and the nucleoprotein, as you can see here. And the nice thing about this study, they follow these people over time to demonstrate that the antibodies that bind to RBD and S2 maintained up to 7 months, whereas the nucleoprotein antibodies, they decline over time. This is the demonstration that perhaps the best markers of exposure to the SARS-CoV-2 rely on the antibodies that bind to the spike protein, mostly RBD and NS2. Obviously, we need to take into consideration that the immune response is quite complex. It's not only the antibody production is the key for protection. We have several players taking place in this response, and what we mostly would like, either with the vaccine or producing, especially with the vaccine, is to induce a memory immune response based on B and T cells. But if memory B cells producing antibodies are the central, is a cornerstone for the immune response, obviously the production of these monoclonal antibodies is a way to go. I'm going to jump into Deng, a paper in Science a while ago that used challenge of macaques to demonstrate how the sequential infection can be avoided by immune response. What they showed is, please bear with me in this right-hand side of the slide. Those animals on this side were re-challenged. You can see that they do not develop viremia collected in the throat or even intestine of these animals. So previous infection is protective. In here, you can see the buildup of this humoral immune response with mounting titers of antibodies against the spike protein. Here is the Brooks lab that published a similar paper. I'm calling you the attention to, again, that re-challenge is not followed by significant detection of SARS-CoV-2. And again, there is a boost in the monoclonal, the antibody production by these animals. Now, let's think about a little bit more about using monoclonal antibodies to treat and prevent COVID-19. So the idea it was early in the course of this pandemic. The first paper published was from the Bosch lab in the Netherlands that isolated antibodies against SARS-CoV-2. Then the Burton lab at Scripps did the same. They were able not only to isolate the monoclonal antibodies, but also demonstrate that they can protect a hamster challenge model. The Nussensweil lab at the Rockefeller in New York also were very successful. Last week, he gave a speech here at the São Paulo State Infectious Diseases Congress and said that those best two monoclonal antibodies he discovered were bought the rights to use them by Bristol Myers in the process of development. Also, the Crow lab have also described the presence of a monoclonal antibody in their system. They also protected animals in vivo, showing that these monoclonal antibodies have promise in use in the clinic. With all these mounting results coming from several different groups, there was a rush in putting forward into clinical programs of development of monoclonal antibodies. And here is the first study that started by Regeneron. They used a cocktail that was mentioned in the previous presentation with two monoclonal antibodies and another one that was used for prevention. So this study kickstarted as early as June 11. Was one of the first clinical trials to test monoclonal antibodies in the clinic. Now there are 21 that I could find in the clinicaltrials.gov of several companies. Here you see that there is about 9 companies who are now in phase 1 clinical trials with different monoclonal antibodies to be used in humans. You can find some familiar names here. Just to mention, Sorrento announced a while ago the kickstart of their clinical trial, and they received a significant investment because of that. But big pharma is also putting their hand into this field, such as AstraZeneca. And here are the phase 2 and forward clinical trials. The BeiGene is starting the phase 2 clinical trial, and there are 3 companies that are neck and neck in their phase 3 programs. The first one is the Vir Biotechnology that was bought by the right to use in a partnership by GlaxoSmithKline. They have their phase 2 clinical trial, phase 3 clinical trials ongoing. We were contacted to take part of this study. Angela Freitas in our group is leading that effort at our department. The Abacilera antibody in partnership with Eli Lilly was mentioned already here. already have results, preliminary results for their phase 2 clinical trial, and they are now in the phase 3 clinical trial. And the Regeneron designed the phase 1/2/3 clinical trials, many of them, and they're ongoing as we speak. We are enrolling patients into this study at our site. So the Eli Lilly presented the preliminary results, which were recently published and presented by in the previous presentation. I'm not going to go into the detail, but just to remind you something that's quite interesting about the study. The outcome was reached significance with the 2.8 grams of the antibody infusion, with a decline about threefold in the ER visits by those who received the antibody infusion. But let me go through a little bit more detail in the Regeneron preliminary results study that we are most involved in. So they announced these results as early as September 29. That shook the field a lot. I mean, a lot of debate about their results. But let me, before diving into some of their results, just call your attention for one thing that was a preliminary analysis they did in the baseline of their participants. So you can see here on the left-hand side that if you look at the virus load in the respiratory tract of those individuals with COVID-19, we're going to find that those who are seropositive have at least 3 log decline in the virus load. It's pretty remarkable, because if you put this in linear scale, look at the median virus load in the seronegatives and seropositives. They go almost to 0. I mean, visually here. And the most interesting is that this finding reflects how early you get someone to with COVID-19. If you get someone early enough, you don't find antibodies being produced by the immune system yet, then the virus load is pretty immense. Therefore, this is probably the basis for using monoclonal antibodies, and potentially plasma has been suggested before, that you need to come early because you can catch someone who did not build their immune response. You can control the virus replication with your intervention. In the results from the Regeneron phase 2 clinical trial go exactly in that direction. Look at those individuals with lower virus load. Here is above 10 to the 4th. You can see that there's no difference between placebo arm, the combination of a lower dose cocktail, and the higher cocktail dose. As you climb up in the virus load at the baseline, then you start seeing a split between how is the virus load behaving over time in those individuals who were in the placebo arm versus the monoclonal antibody arm, meaning that probably the most and the utmost importance is how early you come in with the monoclonal antibody infusion. Therefore, this is a pretty promising approach to treat early infection stage disease. And same thing they saw in the clinical outcome. Those individuals with seronegative have shown a split between the placebo versus the 2 doses of the cocktail, which was not seen in the seropositive and was diluted in the overall population participating in the trial. So Regeneron studies are ongoing. We're participating in the treatment studies, the 2066, in hospitalized patients that restricted to individuals at early stage disease to participate. And the 2067 was just approved to treat outpatients with 1 outpatient infusion of monoclonal antibodies. So you see the names here. Remember the nomenclature. VI is virus, MAB or UMAB is human origin of antibody. So now you can see here very preliminary data from their phase 2 and the first 3 clinical study, reproducing the same finding that those individuals who are positive in the serology have a much lower virus load compared to those who are negative. And this is present all across the volunteers participating to the two studies, either in the outpatient population or inpatient population. Now, this whole field got really, again, into the public debate when Donald Trump developed COVID-19 and received the Regeneron cocktail as a treatment, as a compassive access, or just because he's the president. Come on. And both producers who are more advanced in their programs, Eli Lilly and the Regeneron, had received emergency use authorization by the FDA in the end of November. So the monoclonal antibodies are starting to be available in the United States, especially for early stage treatment of COVID-19. So in summary, we have here a great opportunity to develop new approaches against emerging pathogens. The use in treatment and prevention is wide open. I think the use in treatment is going to be restricted to those individuals at early stage disease, and probably will be a way to prevent hospitalization and development of severe cases. And then there's the prevention, although prevention usage may be quite restricted after the announcement of efficacy results for at least 4 vaccines, but mostly the Pfizer and Moderna vaccines with an unbelievable 95% protection, which is way beyond our wildest dreams when these trials started. And several other vaccines will come into the field and probably take over the prevention of COVID-19. Monoclonal antibodies are well tolerated. The only problem is for those treatment protocols, it's IV infusion, which is a limiting factor, but there is also a push to start using monoclonal antibodies that are more potent in a lower volume and given subcutaneously. And finally, there is the issue of resistance. You saw that Eli Lilly and Regeneron are also testing combination of monoclonal antibodies, obviously to a legit concern about emergence of resistant strains to these monoclonal antibodies. In fact, as personally, I heard from the Regeneron people that in vitro, if you use those monoclonal antibodies isolated in very in diluting concentrations with SARS-CoV-2, you start inducing escape at the lower concentrations, which is a problem. But they never saw escape in the combination of the 2 monoclonal antibodies. Then finally, I'll wrap up by saying that this has opened another field of research, and there are several groups that are looking for the discovery of large spectrum monoclonal antibodies. And here we can even consider an antibody that could not only neutralize SARS-CoV-2, but perhaps all the other beta coronavirus. This approach can embrace not only SARS, MERS, and now COVID-19, treatment of COVID-19, but eventually new emerging beta coronaviruses in the future. I'll wrap up here by thanking our group, especially Angela Freitas, who is leading the monoclonal antibody effort, several of our team members in our clinical site, and a special thanks to Alice Song. I did not present our convalescent plasma results that we finished 2 weeks ago, and but she was mostly involved and coordinated that effort. And thanks to Parexel, especially Fernando, who is coordinating the sites here for the Regeneron trial here in Sao Paulo, and David and George from Regeneron, who invited us to participate. With that, I'll send back to the moderators for discussions.
Thank you very much, Professor Esper. I would like to reiterate that we have opportunities to discussions and questions at the chat in YouTube. Professor Sylvia is receiving the question. We have here some questions. The first one for Professor David, a colleague from Fortaleza here in Brazil, is asking about your current criteria for indicating both convalescent plasma and antibody in treating the COVID-19, if at your institution.
Yeah, so that's a great question. We are directing most of our plasma use to the trial that we're currently running, and I've shown you sort of the details of that. So again, the principles are to try to get people in earlier rather than later. Regarding the antibodies, those are under the FDA emergency use authorization. Those are limited to ambulatory patients and who meet those those criteria of mild to moderate COVID-19. Now, of course, the difficulty is that it is quite difficult to predict who will progress to severe COVID-19 and who of those many people with ambulatory mild COVID-19 will do well without any treatment. And we haven't quite figured that out, I would say.
Thank you very much. And for one question for Professor Esper, it's a question regarding antivirals. If you have any perspective for the use of antivirals.
Sure. I think antivirals are also promising approach to treat early stage COVID-19. I'm very skeptic about using Remdesivir. I did not present the data, but everybody knows the mixed results from different groups. But even those positive clinical trials for Remdesivir have shown a very modest effect in treating, especially hospitalized patients. Now, whether Remdesivir could be used in early stage disease is something out in the open. I think we need more data to demonstrate that. We do have Galidesivir is another antiviral drug that acts upon RDRP. We did a first stage of the clinical trial here together with all 4 other sites here in Brazil. Results are being analyzed and no indication yet whether we should forward to the second step of the study. There are some other antivirals under development, especially oral presentation, orally given antivirals. There is one from Merck, which is an outpatient it's for outpatients, given like a 5-day course antiviral that is still waiting for the results, and several others are underway. So, but again, I think the key in using antivirals, if they're going to have a place in the future in the treatment of COVID-19, is going to be in early treatment. The same way we are testing for monoclonal antibodies or convalescent plasma.
Thank you. And one more question, directly from Sylvia, David, but we would like to know about the health insurance coverage for the antibodies and also for plasma.
I
Did
did
you
not get
understand?
that.
About the health insurance coverage, about who pays for the antibodies there
Sure. I
in
think
USA.
this opens this debate, which is how expensive will be these products, and I'm assuming that potentially the cost will be high. We don't have any approved here or licensed, so I cannot answer that question. But one thing I can say, if you look at the number of companies seeking for monoclonal antibody development in the field of COVID-19 is quite high, which will push the competition. The competition will push the price down eventually. So I think that will facilitate a little bit, but it's going to take a while. I'm not sure about how it would be in the United States, although here there is I foresee a significant barrier due to the cost of these products.
In the United States, Dave?
Yeah, I mean, I certainly am not an expert on healthcare expenditures, but as a sort of observer, we obviously have an enormous problem with drug cost in general, and that's outside of the COVID-19 pandemic, where drug costs are just not adequately regulated, and that effective monopolies exist with really disastrous results for patients. I'm not optimistic that COVID treatments will be an exception to that. I think the prices will remain high, and that will lead to further inequality in our healthcare system than we already have.
Okay, thank you. And one more question. Sorry, anyone? Hi, Fernanda, please.
Hi, I have a question. Thank you both for the talks. Those were great. Just to piggyback on the question about health insurance, Trump had said he was going to pay for every American to get the monoclonal antibodies, but I don't know if that's true. But that's what he said after he got his dose. So my question is actually for both of you. I wanted to see your perspective also on the monoclonal antibodies. So as you mentioned, I mean, it is available here in the United States under the emergency use authorization. I think there were 300,000 doses that were initially distributed. We got some doses here, which of course is limited, and our major limitation to use monoclonal antibodies is having a place to give it and having nursing staff available for the infusion. So my question is, if in an ideal situation, if you had this readily available, would you give priority to transplant patients, both solid organ and hematopoietic cell transplant patients with COVID, to get these monoclonal antibodies?
Yeah, I think the key about monoclonal antibody treatment is the number needed to treat, so the NNT. Because if you, let's say you get a bunch of young guys with positive PCR, a bunch of guys that come with a positive PCR in your clinic and they're previously healthy, you're probably going to need to treat thousands of people to prevent one death. Now, if you select those individuals who are at the very high risk for developing severe disease, then the cost benefit and the number needed to treat is going to be cost effective. So I believe that the examples you gave, the immunosuppressed patients who develop disease, is a good example. Therefore, I would, if I had, let's say, 100 vials of monoclonal antibodies in our clinic, we're probably going to select those very high vulnerable individuals, to give the treatment to them. And again, always reminding that for those who were not suppressed, I would put a limited number of the days after onset of symptoms. But for those who are highly immunosuppressed, then we can be much more loose in that definition, because there is a lot of case series and case reports showing that these individuals can sustain viral replication for a long time, sometimes weeks or months. So in that case, I think a monoclonal antibody is a good use.
Yeah, I would agree with that, and that's actually what we have at our institution currently. Is we have immunosuppression as one of the conditions that would put you higher up on the list, and we have the same logistic challenges that you were describing, Fernanda, about how to give it, where to give it, and again, you want to get to these patients very quickly. So that's I think that's sort of the main challenge, especially now that we're trying to do a lot by telemedicine, and so it's a hard decision to figure out who do you want to bring in to get a quick test, and who are you telling, stay away from the hospital, and that can be challenging, especially when you're looking at these papers, and it really seems that the first sort of 3 days are probably the crucial period if you want to do any good.
Could you please comment some few words about the super infections, bacterial and fungal infections, and how to screen patients, or when to introduce antibiotics or antifungals?
Yeah, I can comment briefly on that. Obviously, there's a lot of mostly observational literature. I would point out one limitation of a lot of the papers, and this is not because people are bad researchers, but it's just a very difficult distinction between respiratory colonization and respiratory infection. The diagnosis of pneumonia, especially in those who are mechanically ventilated, as all of you know, is incredibly difficult, and is a clinical one where you use multiple components, and it's hard to definitively classify somebody as being infected versus colonized. So the studies in which tracheoaspirates or even BAL samples are reported as signs of superinfection, I think do have to be taken with a grain of salt, because of course you have all sorts of things always in your airways, and then especially when you're intubated. I have not really seen a clear signal that any specific bacterial or fungal infection is more associated with COVID-19 than in a comparably ill, prolonged, intubated population. There's been a fair amount of literature on Aspergillus, as I'm sure you're aware. We have not seen that here as much, and it may be geographically dependent. But I would, for now, I would use sort of a general ICU approach, so that if you see people who seem to have a biphasic illness, who develop a new respiratory symptoms, who were first weaning off the vent and then are no longer able to be weaned off, who have increased, those kind of signs of ventilator-associated pneumonia, you would work them up as you would any other patient. I'll stop there.
Yeah, I do concur with everything, and like to add that in the beginning of this pandemic, I was more inclined to use antibiotics at early stage and more aggressively. But as time passes by, I'm becoming less and less enthusiastic to prescribe empiric antibiotics to COVID-19, especially to those cases that are mild or moderate, because many people get afraid when they see the reactive C protein increase and the cough that doesn't go away and those lesions in the lung, and keep giving antibiotics to everyone. But we're much more cautiously about giving antibiotics to these people. But I totally agree that for those that are hospitalized and ventilated, this is a completely different game, and they usually develop secondary infections. We saw Aspergillus cases here quite often, by the way, and I think this is a widespread phenomenon to see secondary infections in these more severe cases.
I have a question for both. Tomorrow we are going to have more data regarding superinfection, but as I don't know if you are going to join us tomorrow, I'd like to ask both if you are using routinely PCT to guide antibiotic use or to stop the use of antibiotic in patients on mechanical ventilation, and also if you are doing galactomannan as routine in those patients mechanical ventilation.
So Sylvia, I usually use C-reactive protein to guide some of these decisions, but never as a single parameter. I mean, just looking at the entire picture clinically and in the lab work. As far as galactomannan, we're not using routinely, no.
Yeah, so at our hospital, we don't have procalcitonin available in-house. So if we want to order it, it's a send out. So by the time you get it back, your decisions have already been made, and it's no longer useful. We do have CRP. I'm not aware of our MICU group or our ICU group using that routinely. I don't think they are as to guide antibiotics. It's much more of a multifactorial decision, as you also mentioned, Dr. Pellis. So the question about galactomannan in general, outside of the hematologic malignancy population, serum galactomannan as just monitoring does not have a great track record, as you know, and it's probably not sensitive enough for that purpose. Where we do use it is in BAL galactomannan, so BAL fluid. There, I think it's a fairly reliable test. Of course, like every test that detects an organism, it won't tell you whether you're dealing with infection versus colonization. But certainly, if there was a patient with a consistent clinical picture who underwent bronchoscopy, and we would certainly get the galactomannan, and if it was positive, even without growth on fungal culture, we would treat as invasive pulmonary aspergillosis.
Yeah, because I'm asking because there in the beginning of pandemic, and a lot of people were worried about the increase of case of an invasive and aspergillus patients with COVID-19 on mechanical ventilation, as happened with an influenza during the influenza outbreak. And they published 2 years ago a paper trying to set up criteria for patients on mechanical ventilation using both galactomannan and blood and also in BLL. But I'm not sure about if it's a good tool to use during the pandemic or not? Because it seems that we don't know the cutoff, the ideal cutoff to use, mechanical ventilation in different situations that's not neutropenic patients. Because of that, I ask you guys your opinions to share with all people that are joining our webinar. Thank you.
Thank you very much. Sylvia, I think we can finish it now. You have some words, something to say before finishing? That's okay. I would like, in my name, in name of Professor Sylvia and all of our group of immunocompromised host patient infections in immunocompromised host patients, thank you very much for the excellent participations. Thank you very much for the audience, and we are here today at the 4:30 p.m. Okay? Thank you very much.
Thank you, David.
Thank you.
Bye-bye.
Bye, Fernanda.
Obrigada, Fernanda. Tchau, tchau.